Day-Tiba Biotech is a preclinical biotech company that spun out of MIT in 2018 and has been developing a library of molecules for RNA delivery. The initial development of our platform occurred at the Whitehead and Koch Institutes by our chief scientist, Justine, who I was lucky enough to work with for several years as part of an undergraduate research opportunity. In addition, Carl Rubing, our CEO, is an alum of MIT. So at Day-Tiba, we are focused on engineering around the many limitations of current RNA delivery technologies. Our platform is referred to as RNable, where ABLE stands for amino alkyl branched lipidoids. Our delivery materials are composed of biodegradable dendrimer molecules with ionizable amines for RNA binding and are modified with terminal hydrophobic tails for self-assembly. Our materials are molecularly defined and range from 2 to 5 kilodaltons in size. So as an example of some of the data that we've generated with these molecules, we have here a benchmark study in mice where we demonstrated higher expression of a reporter gene and lower inflammation following intramuscular administration of our RNable nanoparticles compared to LNPs formulated with MC3. At 24 hours post-administration, concentration of circulating inflammatory cytokines were negligible in the RNable groups, whereas for the MC3-based benchmark groups, there were relatively high concentrations of cytokines such as RANTES, MCP1, and IL-6, which are indicators of inflammation. So this highlights an opportunity for the use of our platform for therapeutic applications. So in addition to the lower inflammatory response to our material unlocks alternate routes of administration, as we can administer our materials intranasally without the adverse effects that are seen with LNPs. The same dose of LNP delivered intranasally induces sickness in mice with higher clinical scores and in some cases lethality compared to Day-Tiba's RNable delivery materials. So our materials have also shown promise for delivery to the heart, an organ that's difficult to access with LNPs. In the experiments here, RNAs encapsulated our RNable materials were administered by IV injection, and 24 hours later, luciferase and IRFP expression were induced in the heart at levels significantly higher than the MC3 LNP benchmark. So on the left here, we have luciferase expression, and on the right, we have IRFP expression in a whole heart. So we have since optimized formulations further using 2 of our delivery materials for higher levels of expression in the heart. We found that the different formulation conditions can make a drastic impact on expression levels in different organs. So we're continuously screening our materials and optimizing formulation conditions to maximize expression in the heart. So to determine off-target effects, other organs were analyzed for luciferase expression, and the primary off-target site of expression was the spleen. Even so, the RNable materials have lower expression than the LNP in the spleen. So through further optimization efforts, we've been able to increase potency and specificity for the heart. But we are very excited to continue building and screening our RNable library of delivery materials and are actively engaging in partnerships to further investigate potential therapeutic applications. So Day-Tiba is working on an in vivo validation of model therapeutic mRNA payloads through early 2023. And we are seeking partners, both domestic and global, with expert knowledge in high-value cardiovascular indications. We are also interested in finding partners with experience with or access to preclinical assessment tools for heart disease. Beyond cardiovascular indications, we're looking to collaborate with groups that are interested in extrahepatic mRNA therapies that may benefit from low inflammatory delivery. So if you're interested in learning more, please feel free to join my breakout room or send me an email. Thank you for your time.
Thanks, Justine. So a couple of questions for you. Have you tested any other organs?
Yes. So when we are doing these experiments, we're taking out a variety of organs and we're imaging them. We have seen some expression in the lung and some expression in the heart and spleen with other delivery materials. What I've shown here is just the lung, but we are taking out a variety of organs.
So connected to that, are you thinking of infectious diseases as well?
So our technology is being utilized both for potential therapeutics and for vaccine applications. So these particular materials we're utilizing for heart delivery or lung delivery, but we have other materials that are performing well as vaccine.
Okay, and one other question. What about IP issues and patents, since there's a lot of contentious happening in that field?
Yeah, so we are separate from the LNP technology that is kind of the state of art for RNA delivery. So the ionizable lipids and polymers are separate from us. We have our own IP. We've been able to aggressively patent our materials.